Ladies and gentlemen, good afternoon. First, I want to thank the jury members for agreeing to arrange this PhD defense via video conference, Professor Yakyaken and Professor Gerardishan. I'm also really grateful for the jury members, my supervisor, Dr. Vincent Cantagrel, and the president, Professor Jean-Louis Guéa, for being here today. This thesis has been conducted in the University of Lille in the unit of Glycobiology structurale et fonctionnelle under the direction of Dr. François Foulquier and Dr. André Klein. It is entitled Insights into TMEM165 function in Golgi glycosylation and lysosomal manganese-induced degradation. To introduce this work, I will first define glycosylation in biology and then focus on N-glycosylation. The structure of the N-glycan will be described as well as the biosynthesis along the secretory pathway in order to discuss the pathologies that appear in case of an impairment of this biosynthesis, such as CDGs for congenital disorders of glycosylation. In a second time, I will present the current knowledge about TMEM165 CDG and TMEM165 protein, especially their role in glycosylation and the link between TMEM165 and manganese. The main result obtained during this PhD will be exposed in order to propose a new model of function of TMEM165 that will be opposed to the current literature. The majority of the dry mass of a cell is composed on 4 different macromolecules: lipids, nucleic acid, proteins, and carbohydrates. Carbohydrates, also known as saccharide or simply sugars, are essential for cell structure, metabolism, and almost all the biological processes. The regulation is carried out mostly by glycosylation, and 2% of the coding genome is dedicated to it. Glycosylation modulates protein and lipid physiochemical properties and function, and we have various types of glycoconjugate primarily defined according to the linkage to the lipid or the protein. We have glycolipids, mucins, glycoproteins, proteoglycans. The majority of the secreted and transmembrane proteins are glycosylated, and over 70% of the eukaryotic proteome is thought to be glycosylated. This explains their role in cell-cell communication, development, immunity, nervous system, and pathogens arguments. This glycosylation is a glycoconjugate is linked with is a protein that carries one or more sugar covalently attached to the peptide backbone, usually a O or an N linkage. O glycosylation is generally the attachment of a sugar to a serine or a threonine residue of a protein. However, in this presentation, we will focus on N glycosylation. The structure of the N glycan starts with a common core in red. It gives birth to 3 different potential structures: the oligomannose type with mainly mannose residues branches, the complex type with branches elongated from N-acetyl lactosamine residue and ended by sialic acid, and the hybrid type that is a mix of the 2 previous ones. And the biosynthesis is conducted through the secretory pathway in 2 main compartments, the ER, endoplasmic reticulum, and the Golgi apparatus, GA. The synthesis of a N glycan starts with the sequential addition of monosaccharide on a lipid molecule, the dolicol, in order to form the LLO, lipid link oligosaccharide precursor. Every single step is conducted by a single enzyme. After a flip-flop process orienting the glycan inside the lumen of the endoplasmic reticulum, the glycan will grow up until it reaches a conventional structure, tetradecasaccharide. At this point, a very complex multi-subunit enzyme, OST, oligosaccharide transferase, will transfer the glycan from the dolichol to a nascent protein. Once transferred, the glycan will undergo various trimming and processing in the ER and in the Golgi apparatus. The Golgi glycosylation consists mostly on the addition of lactosamine, sialic acid, and fucose residue to obtain a mature glycan. We can already appreciate that every single enzyme is important, and any defect in this process will lead to a dramatic biological impact. Indeed, any defect in the state that precedes the transfer by the OST will lead to a disease called CDG type 1. CDG type 1 is the hypoglycosylation of a glycoprotein, a missing glycan, because the allelosynthesis is dysfunctional. A CDG type 2 is the aberrant trimming and processing of a N glycan after it has been attached to the protein. Actually, the majority of the CDG type 2 are due to a defect in the Golgi glycosylation process. The abnormality are either due to a defect or a mislocalization in Golgi enzyme, glycosyl transferase, or nucleotide sugar transporter. And we are now discovering genes that are implicated in Golgi vesicular trafficking and Golgi ion homeostasis with pH and ion. And this was the case for TMEM165. What is a CDG? CDG stands for congenital disorders of glycosylation. It's a group of rare genetic diseases with a mainly autosomal recessive transmission. It has first been described 40 years ago in Leuven, in Belgium, by Professor Jaak Jacken, who is honoring us with his presence today. Today, more than 150 different CDG has been identified, and it can affect all the glycosylation processes. But the biggest group is the one that affects N glycosylation with more than 70 subtypes. Clinically, a CDG is a multi-system disorder with a variable phenotype, even among siblings, and an unpredictable severity. The gold standard method to diagnose a CDG is isoelectrofocusing. Separation of a glycoprotein marker, in this case, transferrin, allows the identification of N glycosylation defect. Transferrin is synthesized in the liver and has two glycosylation sites. In control cell and control serum, the majority of the transferrin is tetracylated with significant amount of penta and hexacyanotransferrin. In CDG type 2, where there is a missing glycan, there is an elevation of D and R-cyanotransferrin. Whereas in CDG type 2, there is an elevation of all the hypocylated forms of transferrin. In 2012, a new protein has been involved in the CDG type 2. It's TMEM165. It belongs to the uncharacterized protein family that is highly conserved throughout evolution. However, its function was completely unknown back then. Using autozygosity mapping, mutation in TMEM165 gene were found in 5 different patients sharing a common clinical picture with psychomotor and developmental delay together with a particular bone phenotype with dwarfism, early osteoporosis, and a severe scoliosis, as you can see on this radiograph. A type 2 CDG glycosylation defect were identified by transferring isoelectrofocusing. Due to the apparent glycosylation defect in this patient, our team first wanted to establish a link between TMEM and glycosylation. Using mass spectrometry in patient fibroblasts and in TMEM165 KO cell line, we can see that there is a high glycosylation defect with accumulation of truncated N-glycan, mainly galactosylated. In our team, we use LAMP2 as a glycosylation marker. LAMP2 has various glycosylation stats, and its migration profile reflects the glycosylation status of the cell. In normal cell, the molecular mass of LAMP2 is around 80 kDa, whereas in KO TMEM cell, when there is a glycosylation defect, the molecular mass is lower. Really interestingly, the addition of 1 micromolar of manganese during 3 days in the culture media was sufficient to suppress the glycosylation defect, as you can see here. So even if the link between TMEM and glycosylation is now completely established, the ion transport of TMEM is still controversial. In 2013, due to a yeast study, the yeast ortholog of GDT1P was first thought to be a calcium proton exchanger in the Golgi of the yeast. Few years later, PAM71, that is actually the plant ortholog of TMEM165, was shown to be a calcium manganese cation antiport transporter that is localized at the thylakoid membrane of the Arabidopsis thaliana, regulating the chloroplastic manganese homeostasis. Few years later, our team first showed that the function of GDT1P will be actually a manganese importer and a calcium exporter that is changing the current dogma. In accordance to this, the first team confirmed the manganese transport of TMEM165 and showed that GDT1P is actually a regulator of the yeast manganese homeostasis. From this observation, a first hypothesis raised. TMEM165 will be a manganese importer at the Golgi apparatus, importing manganese inside the Golgi that are essential for the activity of glycosyl transferase, especially galactosyl transferase. In the absence of TMEM, the enzyme becomes inactive, unable to elongate the building N-glycan, leading to the appearance of hypogalactosylated and hypothylated forms of glycans. If TMEM165 imports manganese, the absence of TMEM will lead to a reduced concentration of manganese inside the Golgi lumen. We use GPP130, a Golgi protein that is sensitive to luminal manganese concentration. As you can see, in TMEM165 depleted cell, there is a reduction of GPP130 manganese-induced degradation. So TMEM165 imports manganese. A second interesting finding, also linking TMEM165 and manganese, is that TMEM is itself degraded under manganese supplementation. It is specific of manganese in the normal cell because it doesn't appear upon supplementation of any other ion. It is also visible in immunofluorescence. Here you have TMEM in red and LAMP2, that is a lysosomal marker. We use chloroquine. Chloroquine is a drug that blocks the acidification of the late compartments, such as lysosome, and thus blocks the degradation of the proteins inside this cell. Using manganese and chloroquine together in control cell, we can see an accumulation of TMEM165 in the lysosomal compartment, meaning that under high manganese concentration in the cytosol, there will be a really specific mechanism that targets TMEM toward degradation. Even if the particular cellular and molecular mechanism of TMEM is not completely deciphered, the link between TMEM and cellular Golgi manganese is now clearly established because of the restoration of Golgi glycosylation by manganese supplementation, TMEM165 manganese-induced degradation, and the absence of GPP130 manganese-induced degradation in the absence of TMEM. The physiological regulation of manganese is complex, and it involves many transporters. If we have a look at the cellular membrane, we can see that there are many importers and exporters of manganese that are either active or passive. But if we have a look more particularly at the Golgi level, we can see that there is only one other protein that is known to import manganese inside the Golgi apparatus. It's SPCA1. SPCA1 is a secretory pathway calcium ATPase1, also known as ATP2C1, and it has a unique site of fixation for both manganese and calcium with approximately the same affinity. The first objective was to establish a link between TMEM165 and SPCA1 to better understand the role of TMEM in the maintenance of Golgi ion homeostasis. To assess this, we used cell lines that were knocked out for SPCA1 and evaluate TMEM165 expression. Really surprisingly, in the absence of SPCA1, there is a nearly complete loss of TMEM165 expression, and it is also visible in immunofluorescence. Overall, we have a loss of more than 80% of the expression level of TMEM in KO-SPCA1 cells. For the loss of TMEM165, we postulated three hypotheses. First, TMEM165 will be, expression will be repressed in the nucleus in KO-SPCA1 cell lines. Second, there will be a degradation by the proteasome from the endoplasmic reticulum of TMEM by the ERAT system due to misfold of the protein, for example. And third, it could be a lysosomal degradation from the Golgi apparatus. We quantify the mRNA of TMEM165 in wild-type cell and KO-SPCA1 cells. As you can see, there is no significant difference between them, meaning that the loss of TMEM will be post-translational. To assess the proteasomal hypothesis, we use MG132, that is a proteasomal inhibitor, and have a look at TMEM expression. As you can see, this drug could not restore any expression of TMEM165 in the KO-SPCA1 cells, meaning that TMEM165 degradation is not probably taken over by the ERAT system. Then, to assess the lysosomal hypothesis, we use chloroquine. Chloroquine that blocks the acidification of the lead compartment and the activity of the protease in this compartment. As you can see, really surprisingly, we could successfully restore TMEM165 expression using chloroquine in KO-SPCA1. And it is also visible with the accumulation of TMEM165 in the lysosomal vesicles with chloroquine, meaning that there will be a continuous degradation of TMEM165 in KO-SPCA1 cells. Additionally, we have proven that this phenomenon is time-dependent. The more we treat by chloroquine, the more we accumulate TMEM165 in the lysosome. So you will ask, if TMEM165 is barely expressed, what about the glycosylation status of the cell? We use two markers, LAM2 and TGN46, and as you can see, there is no difference in the migration profile of these two proteins in KO-SPCA1 cell. So, meaning that there is no glycosylation defect in KO-SPCA1 cells, the remaining amount of TMEM165 in this cell will be sufficient to maintain a correct glycosylation in this cell. Second, we wonder if the two proteins will have a close localization in the Golgi apparatus. We use PLA, proximity ligation assay. In this technique, the two proteins are labeled with markers that interact only if they are close enough. As you can see, in KO-SPCA1 cell, there is a dramatic decrease of the signal. And we can get that the remaining thin line will be actually the background fluorescence. So with this technique, we could successfully prove the protein-protein in situ interaction of these two proteins. And according to the technique, the distance between TMEM and SPCA1 will be around 40 nanometers. To further understand the link between TMEM and SPCA1, we use cell lines expressing different forms of SPCA1 that were modified in their ion transport. We have a wild-type form, we have a form of SPCA1 that only import calcium, G309C, and we have an inactive form that is D742Y clone. Before this, we should assess the activity of our clone. So we use GBP130, that is the Golgi luminal manganese sensor that has been described in the introduction. As you can see, in control cell, as well as in KO SPCA1 cell, complemented with the wild-type form here of SPCA1, there is a continuous degradation of GBP130 in the Golgi lumen. In the other clones, here, here, and here, there is no import of manganese inside the Golgi apparatus, so GBP130 is stable. Let's have a look at TMEM165 expression in this clone. As you can see, only the wild-type form could rescue TMEM165 expression, meaning that the link between those two proteins is not physical, but will be functional. And to complete our study, we needed a clone that imports majority manganese. Q747A is a hyperactive manganese importer. And as you can see, it could successfully restore TMEM165 expression in KOSPCA1 cell. So this is the first evidence of a link between TMEM165 and SPCA1. And also, TMEM165 expression depends on SPCA1 ability to import manganese inside the Golgi lumen. If GBP130 is a Golgi luminal manganese sensor, we can guess that TMEM165 will act differently. We wonder, what are the peptide domains that are implied in TMEM165 manganese-induced degradation? This was the aim of the second article. But first, let's have a look at TMEM165 structure. This 324 amino acids protein has 7 transmembrane domains and is highly conserved throughout evolution. It is easily seen with sequence alignment of TMEM and its orthologs, PAM71 and GDT1P. In gray and black, you can see the conserved amino acid through evolution. And there are especially in 2 domains, in yellow here, the EXGDK and the EWGDR domain that are actually the signature motif of the UPF family. We wonder if all those amino acids have a role in the function of TMEM or the manganese-induced sensitivity of the protein. To assess this, we generated 26 mutants with point mutation in this conserved amino acid. And to study the role of these mutants in glycosylation after transfection into KO TMEM165 cells, we have a look at LAMP2 glycosylation profile for each mutant. As you can see, after 48 hours of transfection, we have a large panel of LAMP2 glycosylation profile. Here, you can see that some mutants were able to restore fully glycosylated form of LAMP2, some were restoring intermediate form of LAMP2, and some were completely unable to restore glycosylation. Really interestingly, the mutants that were unable to restore glycosylation were more likely to be localized in the 2 signature motifs of the UPS family. Therefore, we focus on these 2 motifs, EXGDK and EXGDR motif. As you can see, most of them were unable to restore any glycosylation on LAMP2 profile. And on this graph, you have the under-glycosylated amount of LAMP2 depicted with white bars. So the absence of restoration of glycosylation can be due to three different things. The absence of expression of the mutants, the absence of activity of the protein, and also a mislocalization of the protein. To assess the localization as well as the expression level, we use immunofluorescent again. I'll let you a few seconds to take note of this slide. We have TMEM165 in red and GM130, that is a Golgi marker. As you can see, all of the mutants were effectively expressed. Second, most of them were showing a Golgi localization. Exceptions are made about two of them, the D251 and the S253 mutants. They are presenting a lysosomal degradation, a lysosomal localization, meaning that the absence of restoration of glycosylation in these two mutants is due to mislocalization of the mutants. In a second step, to assess the role of each mutant in the manganese sensitivity, we use supplementation, as we did before, supplementation of manganese in the culture media in the cell after transfection and look at the stability of TMEM165. On the left side, you can see that in the control cell, as well as in the KO cell, complemented with the wild-type form of TMEM165, we have a rapid degradation of the protein after 4 hours and even more after 8 hours. For our clone, we can see that some mutants were showing a partial or a total resistance to manganese-induced degradation. Here, here, and here. If we combine those observations, we can see that the mutants that are unable to restore glycosylation are more likely to be localized in the two signature motifs of the UPF family, here in red. One is cytosolic and one is luminal. Moreover, the two signature motifs starts with acidic amino acid, the E, glutamic acid, and the D, aspartic acid. We can guess that these regions are the cation binding site of the protein, and they will have a role in the cation affinity and or in the pocket conformational change of the protein because they are implied in the glycosylation function of the protein. If we look at the mutants that were showing partial resistance to manganese degradation, there are 5 amino acids that are important. 3 are on the cytosolic side and 2 on the luminal side. However, we've just said that the D251 and the S253 were actually localized in the lysosomal compartment, so the resistance to manganese is due to a mislocalization of this clone. Overall, this is the first motif that will govern the Golgi ion homeostasis of TMEM. So we can say that TMEM165 is a cytosolic manganese sensor. And this is consistent with the previous statement obtained with GPP130 and SPCA1. Moreover, we have identified 2 potential important amino acids for the regulation of the protein, cytosolic threonine and luminal serine. They are both hydroxylated, and they have a role in the manganese-induced degradation of the protein and also in the glycosylation function. We can see that they may have a role in the ion access to the channel, and as they are hydroxylated, they could constitute potential sites of post-translational modification, phosphorylation or O-glycosylation that has to be elucidated. As a conclusion for this article, we have identified the key domain for the function of TMEM165 in glycosylation, the amino acids that are implied in the manganese sensitivity of the protein, and two potential amino acids implicated in the regulation of the protein. Overall, we can say that TMEM165 is a cytosolic manganese sensor. And this is consistent with previous articles. In 2017, our team showed that the manganese-induced sensitivity will be conferred by amino acid between 69 and 172 that has been checked. And the GPP130 manganese-induced degradation is affected by the absence of SPCA1. Indeed, there is a decrease of the manganese inside the Golgi and an accumulation of manganese in the cytosol. GPP130 manganese-induced degradation is also affected by the absence of TMEM for exactly the same reason. And it is easily seen that TMEM165 degradation is not affected by GPP130 because TMEM is a cytosolic sensor. The second conclusion is there is a functional link between TMEM165 and SPCA1. And this is the first link, the first evidence of a link between those two proteins in humans. TMEM165 present depend actually on the ability of SPCA1 to import manganese inside the Golgi lumen. In the absence of SPCA1, there is no glycosylation defect. Whereas in the absence of TMEM, there is a glycosylation. So additionally, we can say that TMEM165 will be one major manganese importer in the Golgi apparatus in humans. Even after 8 years of study about TMEM165, the question about the nature of the counter ion of the protein is still controversial. As TMEM165 import manganese, we need a counter ion. The first hypothesis was TMEM165 will be a calcium importer. Due to phylogenetic study, yeast study, and the symmetry between the conserved motif, it was thought to import manganese inside the Golgi lumen. This affirmation is supported by several publications, in which one was published this year. In this publication, the team expressed truncated forms of TMEM165 in bacteria and showed that TMEM could import both manganese and calcium with a higher affinity for calcium. However, this model is far from being representative of the expression of TMEM165 in this native form in human cells. On the second hand, some teams support the idea that TMEM165 will be a proton extruder. In some particular cell, for example, mammary epithelial cell, TMEM165 is 25-fold more expressed than the other cells. In these cells, actually, during the lactation, lactose synthase requires a lot of manganese to produce lactose. And this reaction generates loads of protons. They have to be extruded out of the Golgi by a protein, and TMEM165 is thought to be this protein. A second publication released this year showed that there is a decrease of Golgi pH in TMEM165 depleted cells. However, again, this doesn't mean that there is a direct link between TMEM and pH. Out of this, we propose a new model of function of TMEM. TMEM165 will be one of the main importers of manganese inside the Golgi lumen in an ATP-independent manner. And according to the tissue, the species, the physiological condition, it could import calcium or proton. And in some very particular condition, it could also import calcium. I hope I convinced you of a strong link between TMEM165 and manganese, and this leads to consider manganese supplementation as a new therapy for TMEM165 CDG. Today, A clinical trial is ongoing and hopefully it will be promising. This work has been given had led to the publication of 4 original articles here. And I want to thank my team, our lab, and all of the collaborators without whom this work would not have been possible.